Yeah. If you got this on your, okay, it says, Dear caregiver, in science class, your student has deemed it important to relearn a crucial targeted concept in the blank unit. And I wrote photosynthesis there for you. In order to do this, they need to take blank minutes. I usually wrote 15, maybe sometimes 20, to restudy and review their notes. All of you should have the study guide attached to it, okay? Now, what I noticed is some people that struggled on the quiz didn't have their study guide completed. So you think that might have something to do with it? I think so. So what I said is you study for 15 to 20 minutes. Once it's completed, have them review and teach the concept. So you should teach your guardian, just like you taught your photosynthesis, the photosynthesis to your guardian, okay? Then they can sign it and add some comments if they want, okay? Then you can retake, okay, and get rid of the grade that you don't want. Does that make sense, everybody? Okay, it's not a bad thing. It happens, okay? It happens. You can track me down and find me an IE. I've been trying to track people down, but it gets to be a lot if there are multiple people that need to retake. If you want to retake and improve your grade, you take this home, study for 15 minutes. The resources are there for you. You have your notes, okay? 15 minutes is a little bit of time, but not really that much, okay? You can even probably get it done during IE, all right? And then just have your parents sign it if you would like to retake. And just let me know, okay? Everybody cool with that? Corbin, do you have something?
No.
Oh, okay. I thought you were raising your hand. All right. So with that, you can put that, hang that up on your fridge, and be proud of yourself. Oh. Yeah, I guess.
Half a point away.
Okay.
From getting a perfect score.
So with that,
50 out of 50.
we're going to dive back into mitosis. You should have your mitosis sheet in front of you. Olivia, I'm just going to grab hers.
Can I have my toes?
Okay. So get this out. You have 30 seconds to find this sheet.
I'd like to use this small.
Here you go.
My toast.
Got it. Oh, what?
It won't stop it.
You just punched that. Sorry. What'd you get?
I just saw you walk up to a random person one day and just slap. Yes, I know it was you because it was you.
Oh, wow.
I know it was you because it was you. Yes, it was you. So how I did that.
You got it. You got a comment for right?
Oh
You guys will know if you are new to this building. Or
That's what the chapter.
Look at
this
beautiful
perfect score.
Like that's
Totally
where
perfect. It's not
he was
one
but then
half a point away.
He was
Okay.
definitely not going.
So you should have this sheet out and be ready to draw. We've gotten through. We got through prophase, or we at least started prophase.
I think we got through it.
Okay.
We're on the third one. I do work chicken fries.
Chicken fries, those are hilarious. Just engulf the whole thing.
Or you could find it.
Oh, wow.
Okay, so we went through interphase. Interphase is, depending who you ask, not technically part of mitosis, okay? It's where the cell spends a majority of its lifespan just carrying out normal cell functions. And towards the end of interphase, it's going to start replicating DNA. Go ahead. DNA needs to be replicated in cells so that the new cells have the same exact DNA. Absolutely, Corbin. Thank you. Okay, then we talked about prophase, and here's what prophase looks like. Prophase is when the chromosomes, or the chromatin, starts to condense into chromosomes. So here's my cell. and the nuclear membrane starts to disappear. So you should have had a nuclear membrane that looks like this. Okay, it's dotted because it's starting to disappear. It's not completely gone yet. Okay, and the chromosomes, the chromatin starts to condense into chromosomes. Again, how many chromosomes are in a human cell? Corbin? 46. Okay, thank you, everybody. We're not drawing 46 just for the sake of time, right? In prophase, chromosomes start to condense.
Why you
Okay.
doing like this, people?
He called on Corbin.
Exactly.
Oh, no.
I thought it was
right.
right.
It looks like a mess.
I know.
Well, I was gonna
There's a
say
line
it too,
through
but
it.
then I saw Corbin raising his
It
hand.
drew it like that yesterday.
Okay,
Stab.
Stab.
Who's waving my hand?
depending on which pictures you see in textbooks and around on the internet, it looks somewhat like a bow tie. Okay, kind of like an X, kind of like a K. Okay, now what you need to remember is that this is the DNA that's been duplicated. All right, so one side of these is exactly the same as the other side. Okay, so this is actually, it's all one chromosome, but it will be split into two identical chromosomes eventually. Does that make sense? So you need to label these. These are chromosomes. CHROMOS.
I mean,
OMES.
in no way to
Chromosomes. Okay? This is the cell membrane. Okay? Remember, this is the nuclear membrane disappearing.
It is going. Yes, what about
I like toes.
my cold fish?
No, I don't.
Yep, we're still in prophase here. I'm just reviewing because some of us did not have this. This is
And then
still
there's the 2 centuries.
prophase. Okay? Then, what's going to start to happen is the centrosomes. I'm just putting them in boxes. Okay?
Exactly. Nice. Wait a minute. What's with the T-pose?
Okay? So, what they're going to do is start to migrate to the poles of the cell.
Oh,
Okay.
yes, that makes
So, these are centrosomes, and inside of them,
is a T.
they contain 2 centrioles inside of them. So the centrosomes and the centrioles are moving to the poles. Okay? Now, it's kind of a lot, but you know there's a lot of cent, C-E-N-T. The centrosomes are what house the centrioles, and they start to move to the poles. And once they move to the poles, okay, so I'm going to now draw them here. They're going to start deploying spindle fibers. Okay. Oops. Oh, no. They're going to start deploying spindle fibers. Okay. Nanotubes that are fibers. And they're going to start doing this, and they're going to begin to start attaching to the chromosomes and to each other.
This is the next
Okay.
phase, isn't it?
It's going to be starting the next phase. This is like the end of prophase here. Those spindle fibers, so I'll just label that spindle fibers.
I'm gonna label them stigmas.
Okay,
That's what they.
that's what's going to do the pulling apart of the chromosomes and the pulling apart and pushing away to make new cells. Okay, so the centrosomes start to migrate to the poles, and then they start to deploy their spindle fibers to attach to the chromosomes and help separate them. This is the end of prophase. We're going to get to metaphase here shortly.
Why would
Questions?
you want to know?
This
Why we
is-
want to know this?
This
Yes.
is
To understand how cells duplicate.
This
Because
is,
cells have to duplicate.
this
What's
is
that?
confusing.
When are we gonna use that though?
You
The
wanna go?
random person, let's see how this thing goes split.
You're right.
Just like,
That's just
why?
split.
When would you have to use it if you weren't going to
Biochemistry. what?
Probably not. Yeah, what if you wanted to go into a science?
Cells.
How much money would our whole class give if we actually put human in math? I would use the pencil step.
All right, well, that's my cue to move on. So, we all here? Got that all drawn? So, this was part of that. This one is a new one. Okay? Next. Okay, we're gonna move on to metaphase. Okay, this is the M part of hip mat. IP on the mat.
No, it's IP on the map.
Yep, IP on the mat. Okay?
Like you pee on the person map.
Oh my god!
All right, so you have your cell.
I know.
Yes.
But I'm not in an annoying mood.
I have a cell.
Me die.
Corbin be like that.
Me die.
Metaphase. Draw your cell.
I don't even know. That would be difficult to say.
Okay, so we have our centrioles at the poles. This is at the left pole.
Can I get a T? No. Too bad they're giving me a T.
Yeah.
No.
Yeah.
What?
I give you a TNT.
What'd you say, Ryan?
T. Because T.
5, 4,
What's T?
3, 2, spider.
Okay, there's your centrosome with your centrioles. Okay, and that was going to deploy the spindle fibers. Before we deploy the spindle fibers, I'm going to make the chromosomes. So during metaphase, see this M?
No.
Okay, M.
Oh yeah, that was you.
Think middle, middle, middle. Okay, that's when the chromosomes start to line up at the middle of the cell.
Oh my God. Oh.
O'Reilly. Auto parts. Ow.
Auto parts.
No, it's auto parts and then a car falls on.
Ouch!
Oh, so squashy. Wait a minute, what did you say, Brian?
What did you say?
I said, O'Reilly Auto Parts.
Uh-huh.
Yes, that is what I said, Auto Parts.
Auto shirts.
Auto parts.
Shut up.
Just FYI, this is going on YouTube.
Auto.
In that case, like and subscribe.
Yeah, in that case, like and
If you like it,
That's the entire operation.
Conversations.
Yeah. I mean,
But you're actually
taking
taking
a video of this?
don't worry, I'll just take a video on my phone and post it on YouTube.
Welcome
Yeah,
to the annoying channel.
Yes.
This class is so annoying.
Now,
Not
that
because of the
was-
teacher, because of the student.
must be
Corbin.
Yeah, you can edit that part out.
No, I will not be. I didn't go to school to be an editor. All right, so now your chromosomes are lined up at the middle. Okay, I've only put 4 chromosomes. All right, now your human cells have 46 chromosomes.
That's too many.
Okay, so we're not doing that. Too many to draw. This is just an example. Okay, now the chromosomes look X-ish, right? Kind of like a bow tie a little bit.
Okay.
What have you? This is one chromosome made up of 2 sister chromatids. Okay, so
I'm just gonna take a video of this on my phone and post it on YouTube.
I don't want to do that.
Yeah.
Oh.
We're so good at this though.
But Kenny just posted it on YouTube.
The chromosomes line up at the middle.
It's not working.
Chromatin.
They are attached at the center.
So chromosomes. I'm just going to show all these videos to my mom to show her all the stupid stuff we do in class. So then she'll ground you.
Okay, the chromosomes are lined up,
We are.
and they are attached in the middle.
Because they're stupid. They just don't want to move. They're
By a
just
central mirror. At the very center
Center.
of the chromosome is a central mirror.
Oh, I
Okay.
thought you were saying central mirror.
Exactly. I accidentally spelled central liar.
Okay, I have to zoom
How do
out.
you accidentally do
Avery, use your glasses.
Yeah, Avery.
Zooming out. I wish they had glasses that you could actually just zoom in and zoom out, just like.
That's called binoculars.
You don't want to wear those everywhere.
Yeah, you probably look really funky going around with a pair of glasses that have huge binoculars on them.
Okay, so at the middle of each chromosome pair, a chromosome pair is a central mirror, is where it's connected. All right? We have not yet put the spindle fibers in, but they're in the middle, or generally in the middle here. Okay? And what's going to start happening is these spindle fibers are going to be fully formed. Okay? And they're connecting.
That's a lot of form.
Okay? To these chromosomes. Some of them are also connected all the way across.
I'm a little more.
Okay? So your spindle fibers need to be connected.
I'm bored. Now my cells just look like a little black shaped and blob now.
Okay?
What do you do with the
Start to
other
be connected to the spindle fibers.
Oh my god, they all
How
are.
do you make
Wait.
it look so pretty?
Why do we need
Yeah,
so many?
mine doesn't
Because he has
does.
a bigger sheet than a,
Why do they build like
it has a whiteboard.
I dare someone to go
Okay.
in there
Now-
and say like this is
Without
Okay, what's gonna happen is
is
they're gonna be start to be pulled across. The nuclear membrane is fully dissolved. It is no longer there, right? You just have your chromosomes in the middle, the central mirror in the middle of the chromosomes. There's a kinetochore that is also surrounding this, but we don't even really need to get into that.
So
Okay,
basically,
the
they're
centrosome with the centrioles is gonna start to pull it apart from the middle. So metaphase, remember middle. So next to metaphase, right middle. Next to metaphase, right middle. Okay? Interphase is where it spends most of its life. Prophase is the beginning of mitosis. Pro is at the beginning of something. Okay, if I'm being proactive, I'm doing it at the start of something instead of at the end. Then metaphase means middle, okay? Doesn't mean middle, but think of the M as middle, all right? And we're gonna move into anaphase. Anaphase. Somebody read to me what happens in anaphase. Go ahead, Sophia.
Each pair of chromosomes separate, move to each pole, and make a U shape.
Make a U shape. Okay, so now what you're gonna do is go into the anaphase portion.
Do I need to draw an entirely new cell?
Yes.
Nice.
You do one every.
Okay.
I've been just putting it all at the top.
So basically, now we're just separating them into two.
Cause I
Nope,
actually feel
not
bad
quite.
for them.
They're being ripped apart limb from limb.
They don't have feelings. Okay, this.
Neither do you.
If this is actually going on YouTube, this is gonna be the worst YouTube video ever. That's
Oh,
gonna
no,
be the
one of
best
my
YouTube
YouTube
video
videos.
ever.
Oh, no.
Like it's
Okay,
gonna be so
now we're at
bad.
anaphase. Think of the A in anaphase. A for apart. So next to anaphase, right apart. They're gonna start to pull these apart. Okay? It's gonna pull sister chromatids apart in like a U shape. So think of it like this. We're gonna start with the spindle fibers.
Hey Janice, can you sing your favorite song?
Where does that come from?
No, it's the song that you wouldn't stop singing last year.
Last year.
Okay, so don't make the spindle fibers go all the way to the middle.
Oh, damn.
Okay, and you can even kind of start to draw these cells starting to split a little bit.
Hey Janice, I'm serious. Sing the song. The
So
one that
big
you
old
wouldn't stop singing last year in Spanish.
Okay,
Mona Lisa.
The one
do you mean?
Oh,
Then-
Oh, that one?
The
yeah,
car
you better
stop.
sing that now.
Sing it right now.
No, why
Yes.
can't you?
I don't know
Because-
the song.
Because I don't know it. You know better than me.
Or
But what if he doesn't know it?
Yes, he does. He,
Hold on, what's
he wouldn't
happening? I
stop.
can't
Yeah, I was swimming it down.
kind of like in a U shape,
Yeah, he would just-
okay, and it could have probably even gone further. All right, so you can see how this chromosome was pulled apart, and now it's got one chromosome over here and one chromosome over here. You might think, well, that's only half a chromosome, right?
No.
Nope, because it duplicated, right? So that was really like two chromosomes kind of connected.
Wait a minute, I just
Alright.
got it right towards.
Wait, did I get it?
They duplicated.
Oh
my Alright.
I can't believe it, Corbin.
Exactly.
Well, I can believe it because he is ultra smart.
When he wants.
Have we had this conversation before?
No. What?
About not every time there's a silence in the room, do you need to make a comment?
No, I don't think we have. talked
No, we
about
definitely
it.
have. We
I've
definitely.
been thinking about
There's
it
no
this
way
whole
we
time.
have it.
Because they're gonna be keys.
Okay,
Maybe
I mean, it's one day you'll learn. It's just a matter of time. Okay, here we go. So there we go. Now anaphase is starting to pull apart. Again, these spindle fibers are starting to pull apart. There are still some spindle fibers that attach from centrosome to centrosome. And what those ones do, they're not pulling, they're pushing each other to the opposite pole. Okay? So I have spindle fibers.
Fibers that spindle.
Oops. What time is it?
What time
It's
is it?
not like-
It's time for lunch.
Spindle fibers that are pulling and spindle fibers that are pushing away from the centrosome.
Wait,
Okay.
really?
Okay.
But pushing's not nice.
A for apart.
But it
Okay,
is
A for
time.
apart. Hopefully you wrote apart. All right. And we're entering the last phase of
Telophase.
mitosis, which is telophase. Now, for this one, don't just draw a cell right away because it's going to be slightly different. Questions on what we have so far?
No.
No, not yet.
Question, Jake.
I might have a question.
Can you read it? Sure.
Do it.
I
Read it
might have a
out.
question.
I'm gonna get it.
So,
Go ahead, Jake.
Awesome. Telophase. Chromosomes uncoil and become chromatin. New nuclear membrane forms. Centriole disappears. Cell membrane enfolds and separates the cells. Two daughter cells form.
Okay, so that kind of went from telophase into what the final stage is, and that's cytokinesis. All right, so let's start with telophase, or telophase. You might hear it as telophase. Telophase, telophase. Okay, so you have over here, you're gonna have, I don't like this marker.
Transform.
Okay, nope, telophase you can think of like two. T for two.
Oh wow.
is that what the cell actually looks like?
So make a giant peanut. Or maybe-
Close.
It looks like a heart.
Okay,
At a
close to a
market.
peanut.
Oh, yeah.
All right, you're gonna start to see them separate.
Mine looks like
I
a
can't draw.
bean from like the 1980s.
Why do you know what that means? What?
Because
All right,
Dad bought it.
So, like it said, the nuclear membrane is starting to reform. There's my reforming nuclear membrane.
Oh my god. And then there's no.
I really want this thing to go into YouTube.
It is.
Like
and
subscribe. Somebody better go shove a
It looks
board
like
up their,
goggles.
over their chest or
I
something,
look
or
like
break it with
Now,
their
I
head.
swear
They
they
don't
don't,
subscribe.
they don't like and subscribe.
Go get your merch at
Now, what this
www.annoyingclass.com.
Yes,
Stop being a party
in
pooper.
their class.
Okay, nuclear membranes reappear. All right, in there. It was read by Jake. Remember, our chromosomes have been pulled apart now, and they're making two completely different cells. They start to unwind, and they remember when the first one interphase in the first stage, they were kind of just chromatin, slightly unwound, and not really condensed into chromosomes. So they're a little bit more, I guess you could call it loosey-goosey. So I'm gonna have two of those blue ones over here, and two of those blue ones over there. Okay, and then I'm gonna do the same with the red.
Wiggle
work.
Wiggle worms.
Okay, remember the comment that we talked about. Okay,
Yes. I drew a wiggle with my right hand.
Are you left-handed? No.
Imagine breaking your.
But it's different when it's this way and then.
chromosomes
What about
starting
Oh.
to decondense
Like, oh my God.
into chromatin. Again, all right? The centrosomes are starting to disappear.
Chromatin.
Oh, no.
They did.
Happened.
And then centrosome is being a box.
So I'm just like slightly erasing it. Centrosome
You have
starting to disappear.
No. Don't make it.
Okay, so we're starting to see two separate cells that are going to be identical daughter cells, right?
Oh. And that all happened in the course of 15 years.
What also is going to happen is in here,
It's a big circle.
what's this big circle going to be? No, this is the nuclear membrane.
The nucleolus.
Yep, so
Nucleolus, yeah.
nucleolus, you don't have to label this if you already know what it is. Nucleolus starts to reappear.
Wait, so it was like a race from the earth as if Thanos stacked his fingers?
Yeah. So, there you have it. That's telophase or telophase. Now, what the final splitting is where the the cytoplasm actually splits into two new cells, and that is cytokinesis. Okay? Now, what you call this here, where it's making its little indent, the split, that is called the cleavage furrow.
Mm-hmm.
Yeah, I heard a giggle out there. Cleavage furrow. That's where it's starting to cleave, actually. So, like, I think of a meat cleaver. Something, yep, right? It's starting to cleave right there. That's what you call it. It's a cleavage point, right? Yep. Okay, it's starting to cleave right there. And what's being formed in the middle, you don't probably have to label this, is actin. All right, and that's what's going to start to form the new cell membrane of the new cells. Okay? Cytokinesis. That's the word that you need to write. Let me see where you should write it.
Yeah, where should we write that?
By the where it says daughter cells form.
Okay.
That's what you need to write. Cytokinesis.
Plasma?
Kinesis.
And that is the actual splitting. So this will split into two new cells and it'll enter interphase again. Cytokinesis. So like think of kinesiology.
Kinesis.
What?
Kinetic.
Think of Arnold.
Kinetic.
Schwarzenegger.
Okay.
I'm just going to think of telekinesis.
Learn the alphabet of Arnold.
Now, this looks, we've been doing this in an animal cell. This looks slightly different in a plant cell. And here's how.
I was gonna ask that earlier.
Okay? At the end, it looks slightly different. Okay? In a plant cell, we have more of a rectangular thing. What it does is it starts to form a new cell wall. Instead of actually splitting, it starts to form a new cell wall. So we have our nuclear membrane.
So basically, instead of being shaped like a peanut, shaped just like a rectangle.
Kind of. Okay? It starts to form a new cell wall. We call that the cell plate.
Do we drink root beer tomorrow?
Nope, Monday. Cell plate. So that will look slightly different. It's not going to have that cleavage. The cellulose is too hard to cleave like that. Okay?
Too hard, but then there's the animal cell that's
Okay, everybody with me there? We made it through IPMAT or PMAT today. Okay, now tomorrow we're going to be dividing into groups and you're going to be making posters based on this. Tomorrow you need to bring colored pencils if you have them, or markers or what have you, any coloring utensils that you have. So write that down right now. Bring that tomorrow. Rob.
If you leave it curl enough, would it actually kind of look like a dumbbell?
A dumbbell?
Recycling
Yeah.
Sure, if you want it to look like a dumbbell.
That's why I drew it. I actually drew it. My dad did it.
All right.
I actually,
They had this funny drawing on to show you how I wanted to beat for fermentation.
it was.
I recycled my test.